This is called a karyotype, and as you know, human beings have 23 pairs of chromosomes, and that gives you a total of 46 chromosomes. This is after lab technicians will break the nucleus open, get the chromosomes, analyze them, and then cut them out in an imaging instrument, which is a computer, and then you can cut them out and organize them, and then analyze them, and make sure that that a fetus, for instance, is going to be viable when born and is going to be normal. In this instance, this is a female. This is a cell. These chromosomes came from the cell of a female, because you can tell by the sex chromosomes. So it's XX. And all the cells in the human body, all the somatic cells and germ cells, have 46 chromosomes. This is a germ cell. This is a drawing of a germ cell. I correct myself. There are 46 total chromosomes in human beings, in every cell of a human being. So 2 pairs of 23 of each individual chromosome. So if we come back to the karyotype, for instance, of chromosome 1, and that's how they're named, then there are 2. And of chromosome 2, there are 2. There are 2 sex chromosomes. This is a female, so it's XX. In this scenario, this is a germ cell. It's going through the process of meiosis. It goes from a diploid of 2 sets of 23 chromosomes to a gamete, to a sperm in this case, and that's haploid, so that's going to be 23 chromosomes. So they're not going to be in pairs, because when this gamete meets with the egg, then it's going to go back to being a diploid, which is going to be a zygote, or a fertilized egg. So coming back here, what happens is it goes through the process of meiosis, and then it halves the cells, and you actually get 4 sperm from one process of meiosis, one gamete. And this process is going to take place in the testes, in the gonads, in the testes for males and in ovaries for females. Now, when the gamete comes and meets an egg, you're back at the 2N, and that egg is going to start to, that zygote or fertilized egg is going to start dividing through mitosis. So each of those divisions is going to be an asexual division, and it's going to give a diploid number of chromosomes of 46 in each cell. And that's going to continue dividing until you have a ball of cells called a blastula. I do want to mention as well that in the cell, in the nucleus, obviously chromosomes are not this neatly organized. They're tangled up, they're wound over one another, and it's not until they begin going through mitotic division that they actually align at the equator and they get pulled by the spindle fibers to form 2 cells from one, and that's during mitosis. But this is after it's been manipulated on an imaging machine. Anyway, so when referring to chromosome 15, the first trait that chromosomes code for is eye color. So that's a gene, that's a section of a chromosome that codes for one protein. And in this instance, this is a male, these are 2 sperm, and they are heterozygous dominant for brown eyes. So in other words, brown is dominant, that's why we signify it with a capital letter, and blue is recessive, so it's with a small letter. And what happens, you have 2 chromosomes for every gene, so you have 2 genes for every trait that's being coded for, and this male is heterozygous, so it's going to have 1 brown-eyed gene, 1 blue-eyed gene. So there's a 50-50 chance that the blue-eyed gene or the brown-eyed gene will be the one that fertilizes the egg to form a zygote. Okay, and let's say that this male has a baby with this female, and this female happens to have blue eyes. So we're trying to think that there's a 50% chance that either one of these sperm will fertilize this egg. And this is some of the work that Gregor Mendel did when he actually was crossing the pea plants, and he was seeing round seeds and wrinkled seeds, and he was actually able to trace it across F1, which is the first generation, to F2 to F3, and he noticed a pattern, and that pattern was complete dominance, which is what this video is about. So what we have is a male on the top of this Punnett square is what this is called, and the female is on the left. And I mentioned that the female has blue eyes, and that means that because this is complete dominance, she's not carrying a brown gene, because if she did, it would completely dominate the phenotype or how that gene is expressed. So she's going to have two blue-eyed genes, because if she had a brown-eyed gene, then she would have brown eyes. So what we do is we drop the B's, move the B's over, and from this we can deduce that a heterozygous brown-eyed parent, I should say, it doesn't matter if it's a male or a female, if they're carrying the gene, the blue-eyed gene, brown-eyed heterozygous parent has an offspring with the homozygous blue-eyed parents, then that's going to be a 50-50 chance that that baby is going to come out having brown eyes or blue eyes.